So, having carried out a series of reactions to form some desired product, the key steps that follow are purifying the desired product and then analyzing it to make sure you've got what you think you've got. One example of a way to purify is a process called chromatography. The name comes from the fact that it was discovered many years ago that if leaves were crushed up in a solvent and put on a piece of blotting paper, that eventually the components would spread out at different rates, and you would see different colored bands on the blotting paper as a function of the time that it diffused through the paper. It turns out that different chemicals will stick to materials in a matrix for different characteristic times, with the result that the diffusion rates through the matrix depend on the chemical identity of the compound. In this technique illustrated on the slide, HPLC for high-performance liquid chromatography, it's essentially the same as the old blotting paper experiment, except in HPLC, a column is typically filled with beads that have some desired chemical functionalization on the surface. So there are many different types of beads commercially available for packing HPLC columns. The reaction products are put into a solvent that is pumped through this column, and so that different components of the solvent elute at different times from the bottom of the column. In general, the more complex the reaction, the larger the number of side reactions, the more entropy is involved, and the greater the need for separating and purifying a product. The way that one detects that one has got what one hopes one has is to have some sort of a detector here. So it could be, for example, a light source that gives rise to fluorescence that's then detected through a filter on a detector. It could be ultraviolet absorption, and it could even be a measurement of just the refractive index of the solvent. Whatever it is, a series of peaks will occur at the detector as the reagents come by. And over here in this experiment, two different detectors were used. Here we were trying to couple a dye molecule to a DNA molecule. The red curve is the ultraviolet absorption of the DNA, and the blue curve is the optical absorption of the dye. What we observed with the dye alone are the peaks labeled dye. There's some other peaks here with the DNA alone, the peak labeled DNA. However, when we carried out the coupling chemistry, in addition to these peaks, we found an extra one in the light absorption as a function of time that products elute off the column, and this was the DNA plus the dye. So having found out at what time in a given solvent this product elutes off the column, one can then make a lot of the reaction mixture and then just collect around this time to obtain a purified product. That's purification. How does one know that one made what one thought one made? Far and away the most useful technique here is mass spectroscopy, and in particular a technique called matrix-assisted laser desorption and ionization time-of-flight mass spectroscopy. The reaction products are put, and maybe the purified product is put in a matrix that sits in a vacuum and has an accelerating potential so that positive ions are pulled into the vacuum chamber. After the ions have been accelerated, they drift in a field-free region so that ions with higher velocity will cross it faster and then fall into a detector. The plot then is a count of the number of ions hitting the detector as a function of drift time. This can be calibrated. One has the problem that one doesn't know whether the molecules are singly, doubly, or triply ionized, but that can be easily sorted out because of the integer relationship between where peaks occur. And so once calibrated then, one can measure with very high precision the atomic mass of the things that were on the matrix in the first place, simply by measuring the time of flight. And this expression here just comes from deriving the velocity from ZeV, the energy in the electric field V is equal to a half mv squared, from which for a distance s the time of flight can be deduced. So here's a rather complex product made in our lab, and this was indeed the predicted mass in atomic mass units, and here you see the expected peak, showing that we had indeed made what we thought we'd made. By itself, though, MALDI just tells you that you got something of the right mass. One really wants to know the structure. Probably the most useful way of doing this, in addition to X-ray crystallography, but certainly more versatile, is nuclear magnetic resonance. NMR relies on the fact that a particle with a spin, typically a proton, but it could be a carbon-13 nucleus as well, has a higher energy when it is aligned against a magnetic field than when it is aligned with the magnetic field. So that's a little spin vector there. The energy required to flip a spin is given by the change in spin, so the magnetic quantum number here would just go from -1 to +1, something called the Magneto-Jerrick ratio, the spin of the proton, and the magnetic field. So in water, using E equals h bar omega, we can calculate a frequency with which radiation would have to be shone on the mixture of protons in a given magnetic field to flip their spin from one direction to another. The result is to achieve a slight surplus over a Boltzmann distribution of spins in the high energy state. Well, it turns out for a proton in water that the resonant frequency is round about 42 megahertz for a field of 1 Tesla. Now, if this was all there was to it, NMR wouldn't be very useful, but it turns out to be exquisitely sensitive to the local environment. Here's an illustration of what an NMR machine looks like. So here's the sample in a test tube. Around it is wound a coil that is driven by the varying radio frequency signal, swept in frequency, and it's between the poles of a magnet. When one starts flipping spins, you absorb energy from this radio frequency field, and this absorption is then plotted as a peak on a plot like this. Now you'll notice this peak, both of these spectra are in fact proton NMR spectra, but the peaks have many different frequencies. Why is this? Well, in a complex structure, so let's just imagine we have some structure like this, and let's put the protons on like this, which are the protons one might be looking at. Then if the protons are in non-equivalent positions, they see different neighboring atoms. If they're in equivalent positions, this isn't true. And each of the neighboring atoms, if they have a spin, also has a magnetic moment associated with it. This, in consequence, changes the local field that our target proton feels and shifts it. And this shift away from what its resonance would be as a free proton is called the chemical shift. So these plots here are plots of the chemical shift in a given field, and the example on this slide is for two compounds, one the desired compound, another in which the reaction hadn't proceeded fully. And you see that protons associated with these rings here are in equivalent environments here in the symmetrical molecule, and so that one has just two peaks representing protons here and here. However, in this molecule down below, where this chain has not been added, these protons are now all non-equivalent, and so now we see 4 peaks. So NMR can tell us about the structure we've synthesized with exquisite precision.